Welcome to the PubMed Daily Review for May 31st, 2026. Today we scan 980 new open access papers across 229 journals. In this video, we'll cover 10 trending research topics and 10 selected papers for detailed analysis. Trend 1 of 10, Diverse Technical Applications and Methods. Papers explore various technical methods, designs, and applications across diverse scientific and engineering fields. This cluster covers 104 papers. Representative work includes a two-step tilt compensation method for off-axis holographic displays, influence of restorative material types on 3D printed aligner accuracy using intraoral scanners, and integrated multi-omics and WGCNA analyses reveal pathways and candidate genes associated with branching flower development in Nymphia prolifera, focusing on hormone homeostasis and flavonoid biosynthesis. Trend 2 of 10, Cancer Therapy and Mechanisms. This trend explores various approaches to cancer treatment, including novel targets, drug mechanisms, side effects, and underlying disease pathways. This cluster covers 97 papers. Representative work includes new targets in anticancer therapy, massive bleeding from a Bricker ileal conduit during FOLFIRINOX therapy in non-cirrhotic portal hypertension, suspected oxaliplatin-associated sinusoidal injury, and presumed irinotecan-associated conduit mucositis, a case report, and depression among women with cervical cancer. A systematic review and meta-analysis of available evidence. Trend 3 of 10, Neonatal and Infant Health. Advancing understanding, diagnosis, and treatment of health conditions affecting newborns and infants. This cluster covers 51 papers. Representative work includes liver X receptor agonists enhances intestinal repair in neonatal piglets with massive bowel resection. Development of IgA autoantibodies in newborns with atopic dermatitis. Diana protocol of a prospective, non-interventional, observational birth cohort, and assessment of survival and neonatal morbidity rates of very low birth weight infants in Turkey. Turkish Neonatal Society Secrets, TNS Secrets, Study. Trend 4 of 10, AI and Advanced Analytics Applications. Applying AI and advanced analytics across diverse fields for complex problem-solving, optimization, and insights. This cluster covers 67 papers. Representative work includes machine learning-based approaches for structure-activity relationship analysis of heparinase inhibitors, a hybrid transformer PPO framework for multi-objective energy management in renewable-based microgrids, and synergistic tension among the music and lyrics of late romantic choral songs. Trend 5 of 10, Public Health and Medical Education. This trend explores the intersection of medical education, public health initiatives, and healthcare training to improve population well-being. This cluster covers 141 papers. Representative work includes developing content weights for the 2026 Sports Medicine Blueprint, impact of integrating body and organ donation education into anatomy, teaching on medical students' concepts of respect for life, a mixed methods cluster randomized trial, and preventability of severe maternal morbidity, a scoping review of the literature in high-income countries. Trend 6 of 10, Neurology and Brain Health. This trend explores diverse aspects of neurological function, disorders, diagnosis, and therapeutic interventions for brain health. This cluster covers 95 papers. Representative work includes theoretical analysis of low-power synergistic sono-optogenetic control of calcium-dependent synaptic plasticity, prolonged undiagnosed prodromal symptoms, and effect of repetitive transcranial magnetic stimulation combined with lower limb rehabilitation robots on knee hyperextension in stroke patients with hemiplegia. Trend 7 of 10, Anesthesia and Critical Care. Research improving patient outcomes through advanced anesthesia techniques, critical care management, and training. This cluster covers 144 papers. Representative work includes opioid-sparing and opioid-free anesthesia, a review of clinical evidence and implementation considerations, comparison of the mNutric score and the PNI for predicting 30-day mortality in geriatric patients in the ICU, and association between lactate-to-albumin ratio and all-cause mortality in critically ill hepatic failure patients with sepsis. A retrospective cohort study. Trend 8 of 10, Cardiometabolic and Liver Diseases. Research explores prevention, diagnosis, and management of interconnected cardiometabolic and liver diseases through diverse clinical and genetic approaches. This cluster covers 88 papers. Representative work includes a culturally adapted online Mediterranean diet intervention for metabolic dysfunction-associated steatotic liver disease, TEMA study protocol for a randomized controlled trial, from glucose control to cardiometabolic protection, Implications of the 2026 NICE update for clinical practice, and sexual dysfunction and related factors in adult males with familial Mediterranean fever. Trend 9 of 10, genetics and molecular mechanisms. This trend explores genetic factors and molecular mechanisms influencing biological traits, diseases, and regenerative processes. This cluster covers 82 papers. Representative work includes cross-population meta-analysis and colocalization analysis reveal novel and pleiotropic genes associated with rib and teat number in pigs, identification and validation of autophagy-related genes in keratoconus and their correlation with immune infiltration, and mesenchymal stem cell-derived extracellular vesicles for osteochondral defect regeneration. A systematic review and meta-analysis of preclinical studies. Trend 10 of 10, infectious disease and gut health. Investigating infectious diseases and their intricate links with gut health, encompassing microbial roles and host resistance mechanisms. This cluster covers 111 papers. Representative work includes factors associated with the costs of treating privately insured adults with complicated urinary tract infections, occupational risk of gastrointestinal Helicobacter pylori infection among medical and dental staff, a systematic review, and Plant RG, a comprehensive and user-friendly database for plant resistance gene analogs, RGAs. Up next, our first paper out of 10, prevalence and long-term outcomes of brain fog and cognitive impairment in individuals with long COVID. A systematic review by Ahmar Fasih and Abida Praveen from Department of Dentistry, Islamic International Dental College, Islamabad, Pakistan. This paper was selected because it provides a critical systematic review and meta-analysis on brain fog and cognitive impairment in long COVID, directly addressing a major, emerging global health crisis with significant societal and individual impact. Let's break down its key contributions. Long COVID is a challenging condition affecting millions, and two of its most debilitating symptoms are brain fog and cognitive impairment. These issues significantly impact quality of life, yet we still lack a clear understanding of how common they are and what factors influence their presence. This systematic review aimed to shed light on these critical questions, providing a comprehensive overview of the current evidence. To answer these questions, researchers conducted a systematic review of 47 studies, encompassing over 25,000 patients with long COVID. They meticulously extracted data on patient demographics, follow-up duration, and key variables like gender and vaccination status. The team used robust methods, including the new Castle-Ottawa scale, to assess the quality of the included studies and ensure reliable findings. The findings were quite striking. The pooled prevalence of brain fog was found to be 30%, and cognitive impairment affected 25% of long COVID patients. Interestingly, female patients consistently showed higher rates of both conditions compared to males. The study also revealed that prevalence increased with longer follow-up, suggesting these symptoms can persist or even emerge later in the course of long COVID. This systematic review underscores that brain fog and cognitive impairment are not just anecdotal, but are common and persistent challenges for individuals with long COVID. The identified differences across subgroups, like gender and vaccination status, provide crucial insights for risk assessment and personalized care. Moving forward, it's vital that we invest in research to understand the underlying mechanisms and develop effective interventions to improve the lives of those affected. Up next, our second paper out of 10, Detecting Dataset Bias in Medical AI, using a generalized and modality-agnostic auditing approach by Nathan Drenkow and Matthias Unberath from the Johns Hopkins University, Baltimore, Maryland, USA. This paper was selected because it introduces a generalized and modality-agnostic auditing approach to detect dataset bias in medical AI, addressing a critical and pressing challenge for the safe, equitable, and effective. Let's break down its key contributions. Artificial intelligence has shown great promise in healthcare, but it also comes with significant risks. A major concern is that AI models learn from associations, meaning any biases present in the training data can be amplified and hidden during testing. This can lead to unexpected and potentially harmful behavior in real-world applications. We urgently need new tools to uncover these hidden biases and ensure AI systems are fair and reliable. Our study introduces G-Audit, a novel, modality-agnostic approach designed to audit datasets for bias. This method automatically quantifies shortcut learning risks by analyzing how different data characteristics interact. We applied G-Audit to a diverse set of medical AI datasets, including images, text, and tabular data. This allowed us to test its broad applicability across various machine learning tasks. Our results demonstrated that G-Audit successfully identified potential shortcuts and latent biases within various medical AI datasets. We showed its broad applicability across different data types, including images, text, and tabular data. Crucially, the method uncovered biases that were commonly overlooked by traditional qualitative assessment techniques. This highlights GAudit's effectiveness in revealing hidden issues that could impact AI performance. This research offers a critical new tool, GAudit, for ensuring the reliability of medical AI. By proactively identifying dataset biases and shortcut learning risks, we can prevent AI models from making flawed decisions. This is vital for building equitable and trustworthy AI systems that truly benefit patients. Moving forward, integrating such auditing approaches will be essential for the responsible development and deployment of AI in healthcare. Up next, our third paper out of 10, CD40.HIVNV, an antibody-mediated vaccine, induces long-term and recall immunogenicity in non-HIV-1 infected volunteers by Yves Levy and ANRSVRI06 study group from INSERM U955, IMRB, Team Levy, Hôpital Henri Mondor, Université Paris Estate Créteil, 51, Avenue Maréchal de Latreille, Delaware, Tassigny, Créteil, France. This paper is selected as the top choice because it reports initial human trial results for a novel antibody-mediated HIV vaccine, addressing one of the most pressing global health challenges with a potentially transformative approach. Let's break down its key contributions. HIV continues to be a major global health crisis, and finding an effective vaccine is paramount. This study aimed to address a critical question. Can a novel vaccine, CD40.HIV-NV, not only induce long-lasting immunity, but also effectively recall those responses with a late boost in individuals not infected with HIV? Understanding this is vital for developing durable protection against the virus. Researchers initially enrolled 72 HIV-uninfected volunteers to test the CD40.HIV-NV vaccine. After the initial phase, 45 of these volunteers were randomized to receive a late boost of the vaccine, either with or without an adjuvant. The primary outcomes measured included the levels of ENV-specific IgG antibodies, functional CD4+ T cells, and neutralizing antibody titers. Participants were followed for up to 24 weeks after this late boost to assess the durability and recall of their immune responses. The results were quite promising. We saw that IgG response rates against vaccine-matched GP140 antigens reached a remarkable 100% by 24 weeks post-late boost, and heterologous responses were also very high, between 82 to 100%. Neutralizing antibody titers against a key HIV strain significantly increased, from 14 to 22% to 35 to 59%. Importantly, polyfunctional ENV-specific CD4+ T cells also increased and persisted, indicating a robust cellular immune response. The vaccine was well tolerated, and there were no significant differences between the adjuvant and unadjuvanted groups. This study demonstrates that the CD40.HIV-NV vaccine is not only safe, but also highly effective in inducing and recalling broad and potent anti-HIV immune responses. A significant finding was that the late boost successfully recalled immunity even without an adjuvant, which could simplify future vaccine development. While these results are very encouraging, the next crucial step is to evaluate the actual protective efficacy of these immune responses in preventing HIV infection in a larger clinical trial. Up next, our fourth paper out of 10, multi-omics, a sequential workflow for genomic, transcriptomic, proteomic, and phosphoproteomic profiling from biopsy scale samples by Shi Feng and Dongxue Wang from Department of Chemistry, School of Science, Southern University of Science and Technology, Shenzhen, China, International Academy of Pharmaceutical Medicine, Guangzhou, Guangdong, China. This paper was selected because it introduces a highly novel and technically advanced multi-omics workflow that addresses a critical bottleneck in biomedical research, enabling comprehensive molecular profiling from limited clinical samples, which has broad implications for. Let's break down its key contributions. Researchers often face a significant challenge: how to get a complete picture of a cell's biology, its DNA, RNA, and proteins, from just a tiny sample. Existing methods struggle to provide a balanced recovery of all these crucial molecules, making comprehensive analysis difficult. This limitation hinders our ability to conduct in-depth systems biology and translational research, especially when working with precious biopsy-scale specimens. To tackle this, researchers developed multi-omics, a novel phenol-free workflow. They systematically benchmarked this technique to ensure it could sequentially isolate DNA, RNA, and proteins from a single sample. The workflow was rigorously tested across a wide range of cell inputs and various mouse tissues, demonstrating its robustness and broad applicability in a controlled laboratory setting. The multi-omics workflow delivered impressive results. It consistently identified over 8,000 proteins from small samples, demonstrating remarkable depth. Crucially, it achieved strong quantitative reproducibility, with an R value exceeding 0.95, ensuring reliable data. The method also preserved the high quality of DNA and RNA for downstream sequencing, performing robustly across a wide range of cell inputs. This means we can now get a much more complete and reliable molecular picture from even tiny biopsies. This new multi-omics workflow represents a significant leap forward for research. It offers a standardized, practical, and cost-effective way to perform comprehensive multi-omics analysis, especially crucial for limited biopsy samples. While validated in mouse tissues, its potential for human translational research is immense. This robust tool will undoubtedly accelerate our understanding in systems biology and pave the way for new discoveries from previously challenging specimens. Up next, our fifth paper out of 10. Combined treatment of TROP2-targeted CAR-T and vascular disruptor CBP enhances anti-tumor activity in triple-negative breast cancer by Yizhu Chun and Li Zhu from Department of General Surgery, the First Medical Center of Chinese PLA General Hospital, Beijing, China. Key Laboratory of Carcinogenesis and Translational Research, Ministry of Education, Anesthesiology, Peking University Cancer Hospital and Institute, Beijing, China. This paper was selected because it addresses the critical challenge of improving CAR-T cell efficacy in solid tumors, specifically triple-negative breast cancer, by introducing a novel combination therapy that enhances tumor infiltration, representing a significant advancement in. Let's break down its key contributions. Today, we're diving into a fascinating study exploring a new approach for triple-negative breast cancer, a particularly challenging form of cancer. While CAR-T cell therapy has revolutionized some cancer treatments, it often struggles with solid tumors due to poor infiltration. This research investigates whether a vascular disrupting agent, CBP, could pave the way for CAR-T cells to work more effectively against these tough tumors. It's a crucial question, as finding better treatments for TNBC could significantly improve patient outcomes. The researchers employed a comprehensive approach, starting with bioinformatics and in vitro experiments using breast cancer cell lines to confirm TROP2 expression and CAR-T cell function. They then moved to in vivo mouse models of breast cancer to evaluate the combined therapy's effectiveness. The primary goal was to assess the anti-tumor therapeutic effect, the safety profile, and the underlying biologic mechanisms of this novel combination. This robust design allowed them to thoroughly investigate their hypothesis. The findings were quite compelling. First, they confirmed that TROP2 was indeed widely expressed in breast cancer cells, making it a suitable target. Their constructed CAR-T cells showed high cytotoxicity in lab dishes. Most importantly, in the animal models, the combination therapy of CAR-T and CBP significantly enhanced the anti-tumor effect compared to either treatment alone. Crucially, this enhanced efficacy came with a favorable safety profile, as no significant organ damage was observed. Mechanistically, this improvement was linked to increased immune cell infiltration and the upregulation of immune-related factors within the tumor. This study offers exciting insights, demonstrating that TROP2 is a viable target for CAR-T therapy in breast cancer, and that combining it with CBP can significantly boost its effectiveness while maintaining a good safety profile. However, it's important to remember these are preclinical findings, so the next critical step will be to move into human clinical trials to validate these promising results. Ultimately, this research paves the way for a novel and potentially highly effective therapeutic strategy for patients battling triple-negative breast cancer, offering new hope for a challenging disease. Up next, our sixth paper out of 10, Generation of Therapeutic T-Cells from Human-Induced Pluripotent Stem Cells by Bo Wang and Shin Kaneko from Shin Kaneko Laboratory, Department of Cell Growth and Differentiation, Center for iPSC Research and Application, CIRA, Kyoto University, Kyoto, Japan. This paper, despite being a review, outlines a highly impactful and novel research trajectory in generating therapeutic T-cells from iPSCs, addressing a critical barrier in cancer and chronic infection treatment with the potential for scalable, off. Let's break down its key contributions. T-cell exhaustion is a major hurdle in treating diseases like chronic infections and cancer. This paper explores how induced pluripotent stem cells, or iPSCs, offer a groundbreaking approach to create rejuvenated T-cells that could overcome this challenge. It's a really exciting area of research with huge potential for patients. This review paper outlined a comprehensive research program focused on developing therapeutic T cells from iPSCs. It detailed the evolution from autologous to allogeneic off-the-shelf strategies, covering advanced manufacturing protocols and sophisticated gene editing techniques. The goal was to create highly effective and safe cell therapies for a range of diseases. The research successfully established clinical-grade, xeno-free manufacturing protocols for iPSC-derived T cells. They also integrated advanced gene editing for functional enhancement and to reduce immunogenicity. Furthermore, the team optimized synthetic receptor architectures to minimize GvHD risk and even generated regulatory T cells for autoimmune disorders. These are significant advancements in the field. This review highlights the immense potential of iPSC-derived T cells as a universal, off-the-shelf therapeutic. While the progress is remarkable, we still face hurdles like ensuring long-term safety and navigating clinical translation. Future work will undoubtedly focus on refining these technologies and bringing these innovative therapies to patients. Up next, our 7th paper out of 10, neurodegenerative and inflammatory biomarkers are detectable in saliva and multiple sclerosis by B. Castelli and Una Fitzgerald from CRAM, Research Ireland Centre for Medical Devices, Galway, Ireland. This paper was selected because it introduces a highly accessible and non-invasive method for detecting neurodegenerative and inflammatory biomarkers in saliva for multiple sclerosis, directly addressing a critical clinical need for improved. Let's break down its key contributions. Multiple sclerosis, or MS, is a complex neuroinflammatory and neurodegenerative disease. Current methods for diagnosing and monitoring MS, such as MRI and lumbar punctures, are often expensive and invasive for patients. This creates a significant need for more accessible and less invasive diagnostic tools. Our research aimed to explore whether key biomarkers associated with MS could be reliably detected in more easily obtainable biofluids, like saliva. We conducted a case control study involving 37 individuals living with multiple sclerosis and 23 healthy controls. Our team collected various biofluid samples, including serum, saliva, nasal secretions, and tears, from all participants. We then measured the levels of several known neurodegenerative and inflammatory biomarkers, such as NFL and CAP-FLC, in these samples. The primary goal was to determine if these biomarkers were detectable and differed significantly between the MS group and healthy controls, particularly in less invasive samples like saliva. Our findings revealed that all the biomarkers we investigated were measurable in both serum and saliva samples. Importantly, we observed significantly higher levels of neurofilament light chain, NFL, in saliva, with a p-value of 0.03. Similarly, kappa-free light chains, CAP-FLC, were significantly elevated in saliva, showing a strong p-value of 0.003. We also found significantly higher levels of chitinase, 3, like 1-Chi3L1, in saliva, with a p-value of 0.021, indicating their potential as indicators of MS activity. This study highlights the exciting potential of saliva as an easily accessible and non-invasive biofluid for detecting key biomarkers associated with multiple sclerosis. While these findings are promising, it's important to remember this was a cross-sectional study with a relatively small sample size. Therefore, the next crucial step is to conduct larger, longitudinal studies to validate these results and establish the clinical utility of these salivary biomarkers. Ultimately, this research paves the way for developing more patient-friendly and cost-effective methods for MS diagnosis and monitoring. Up next, our 8th paper out of 10, a pegylated nanobody against HBsAg exhibits pleiotropic antiviral efficacy in hepatitis B models by Kun Yang and Xue-Fei Kai from Key Laboratory of Molecular Biology on Infectious Diseases Ministry of Education, Chongqing Medical University, Chongqing, China. This paper was selected because it introduces a novel pegylated nanobody with a pleiotropic antiviral profile against hepatitis B virus, demonstrating significant advancement towards a functional cure for a global health burden. Let's break down its key contributions. Hepatitis B remains a significant global health challenge, with current treatments often falling short of a functional cure. This creates a pressing need for innovative therapeutic approaches. Our research aimed to explore a novel strategy to address this persistent viral infection. We started by isolating a high-affinity nanobody, VHH4917, from an alpaca phage library, targeting the HBsAg. We then engineered this nanobody using site-specific pegylation to improve its properties. The efficacy of this modified nanobody was rigorously tested in mouse models of chronic HBV infection. Our findings were very promising. We observed that the nanobody accelerated HBsAg clearance in living systems. Crucially, it also potently suppressed key aspects of HBV activity, including replication, transcription, and particle secretion. Furthermore, Epigenetic significantly extended its circulating half-life and boosted its antiviral efficacy in vivo. This research highlights VHH4917 as a promising new therapeutic candidate for chronic hepatitis B, showcasing its broad antiviral capabilities. While these results are exciting, it's important to remember that these findings are from mouse models, and further human studies are essential. Ultimately, this work paves the way for developing innovative nanobody-based immunotherapies to tackle this challenging disease. Up next, our 9th paper out of 10, polypeptide-engineered lipid nanoparticles for mRNA delivery with limited immunogenicity by Jinyue Zheng and Ian Yang from Bioprocessing Technology Institute, BTI, Agency for Science, Technology, and Research, ***, 20 Biopolis Way, the Centros #06-01, Singapore, Singapore. This paper was selected as the top choice because it addresses a critical and widespread limitation in mRNA delivery technology by introducing a novel, non-immunogenic lipid nanoparticle component, which has the potential to significantly enhance the safety and. Let's break down its key contributions. Lipid nanoparticles, or LNPs, are crucial for delivering mRNA therapeutics, but they come with a significant challenge. Many current LNPs use a component called PEG, which can trigger an immune response, leading to anti-PG antibodies. This can cause hypersensitivity and reduce how well the treatment works over time. So, there's a real need to find new, safer ways to deliver mRNA without these immune side effects. To address the immunogenicity issue, researchers designed and screened new poly-D-L-serine (PDLS) lipids as alternatives to PG. They systematically evaluated various PDLS lipid structures to find the optimal ones for forming stable LNPs with high mRNA encapsulation and transfection efficiency. These new PDLS LNPs were then rigorously tested in cell lines and mice to assess their mRNA delivery capabilities, immune responses, and overall safety compared to existing formulations. The results were quite impressive. The newly developed PDLS LNPs showed superior mRNA delivery and elicited robust cellular and humoral immune responses in mice, outperforming the clinically approved BNT162b2 formulation. Crucially, repeated dosing with these PDLS LNPs triggered only minimal anti-PDLS IgM production, a stark contrast to their PEG-based counterparts. Furthermore, these innovative LNPs proved to be highly stable, maintaining their integrity during frozen storage for over 6 months, with no observed systemic toxicity. This research marks a significant step forward in mRNA delivery technology. The development of PDLS LNPs provides a promising, immunologically inert alternative to current PEG-related systems, potentially overcoming issues of hypersensitivity and diminished efficacy with repeated use. This success underscores the power of rational design in creating safer and more effective biomaterials. Looking ahead, the next critical steps involve validating these exciting findings in larger animal models and ultimately progressing to human clinical trials to bring this innovation closer to patients. Up next, our 10th paper out of 10, Apatinib enhances anti-PD-1 efficacy by inhibiting exo-70-mediated exosome secretion in pMMR/MSS colorectal cancer by Lingying Zhao and Tian Wei, who from Xiamen Key Laboratory for Tumor Metastasis, Cancer Research Center, School of Medicine, Xiamen University, Xiamen, China. This paper was selected as the most impactful because it directly addresses a critical and pressing challenge in oncology: overcoming immunotherapy resistance in proficient mismatch repair/microsatellite stable colorectal cancer, a patient population that. Let's break down its key contributions. Colorectal cancer remains a significant global health challenge, marked by high morbidity and mortality. While immune checkpoint inhibitors have revolutionized cancer treatment, a large subset of patients, specifically those with pMMR/MSS colorectal cancer, unfortunately see very limited benefits. This presents a critical unmet need in oncology. Our study aimed to investigate if combining Apatinib with an anti-PD-1 therapy could overcome this resistance and improve outcomes for these patients. We conducted a single-arm, exploratory clinical study to evaluate this combination in patients with advanced metastatic colorectal cancer who had exhausted other treatment options. The intervention involved camrelizumab, an PD, one agent, combined with ipatinib. Alongside this clinical investigation, we performed mechanistic in vitro experiments to understand how ipatinib might be influencing the tumor microenvironment and immunotherapy response. Our primary outcomes included assessing the clinical efficacy and uncovering the underlying biological mechanisms. Our clinical findings were very encouraging. Ipatinib significantly enhanced the therapeutic efficacy of immunotherapy in these challenging pMMR/MSS colorectal cancer patients. Delving into the mechanisms, we discovered that ipatinib improved outcomes by modulating the tumor microenvironment. Specifically, our in vitro experiments revealed that ipatinib reduced levels of exosomal PD-L1 by inhibiting tumor-derived exosome secretion, a process we found to be mediated through the regulation of exo-70. These results provide a strong rationale for this combination. This study offers a significant step forward, suggesting that combining ipatinib with camrelizumab could be a viable strategy for pMMR/MSS colorectal cancer patients who currently face limited treatment options. While these results are exciting, it's important to remember this was an exploratory single-arm study, and larger, randomized trials are essential to validate these findings. Crucially, we've identified a novel mechanism. Targeting exosome secretion via exo-70 could be a key to overcoming immunotherapy resistance, opening new avenues for future research and therapeutic development. That's all for today's PubMed Daily. If you found this helpful, please like, leave a comment, and subscribe for daily paper summaries. See you tomorrow.